Hello, everyone, and welcome to today's webinar, Recent Advances in Covalent and Non-Covalent Drug Discovery with TMT Quantitative Chemoproteomics. My name is Delia Osborne with LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. We encourage you to participate today by submitting any questions that you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as time allows. At the end of the presentation, we also will be addressing any technical issues should you have trouble seeing or hearing the presentation. I'd like to now welcome our speakers, Alexey Chernobrovkin, Ph.D., and Qing, Ph.D. For a complete biography on our speakers, please visit the presenter window at the top of your screen. I'd now like to turn the floor over to Qing.
Good morning, everybody. Thanks for joining me this webinar, and thanks Thermo for organizing this opportunity for us to share some of the research we're doing. So to start with, this is a TMT based drug discovery seminar or webinar. I do want to refresh everybody in terms of what TMT is. So tandem mass tags are these kind of chemical barcodes that nowadays allow us to multiplex up to 35 different samples and analyze everything simultaneously. So they all have this the same structure that consists of a mass reporter, a base group, and also a amine reactor group that will label all the primary amines, peptides, like the peptide N-terminal, as well as lysine side chains. So they all share the same structure. However, by placing like 9 extra neutrons at different places, you can actually create 18 different versions of this reagent. So for example, if you put all the 9 neutrons here, you have a reporter with mass 126. If you move 1 neutron over, you have a reporter with mass 127. You can keep doing until you move all the neutrons to the other side of this line. Now you have a reporter with mass 135. And imagine these are triplet peptide digests from 18 different samples. You can label each one of them with one of these tags, and they will create identical mass shape at 304 Dalton. And the beauty of this technique is you can then pull everything together in a single tube instead of analyzing them individually. And all these signals or peptides, as they are isobaric, they will stack up as a single peak in your MS1 scan where you detect them. You can select this MS1, fragment it to collect MS2 spectra, so you can use it for identification. And again, all the 18 signals are still stacking here. And by further fragmenting of a few of these fragment ions to release the reporter, then you can use this relative abundance of these reporters to quantify protein abundance across all these multiplex samples, right? So sample multiplexing allows us to multiplex 18 different samples to improve our throughput. And also, since we're analyzing everything together, we kind of eliminate all this experimental variability. I'm sure many of you have seen this figure or plot or image many times, right? That just highlights the challenges we're facing in drug discovery. So despite hundreds of years effort, nowadays we're still only be able to barely drug less than 20% of the human proteome, and that's even a smaller fraction when you talk about disease-relevant proteins, right? That's largely because we don't know what binding pockets exist in all these human proteins or disease-relevant proteins. And in fact, in the last 15, 20 years, the people are really getting interested in using this chemical or covalent probes to kind of assess or map out these elusive binding pockets on all the human proteins as an effort to facilitate drug discovery. Because once you know the binding pockets, you can probably design better molecules that can fit that pocket. Just so you know that covalent molecules or also covalent drugs are a viable modality to treat many different types of diseases for over 130 years. It started about 1899 when aspirin and then later penicillin were discovered, right? And these are all classic covalent drugs we're still using today. And you can appreciate in the last few years, there are many of these covalent drugs that are approved by FDA to treat different diseases. And most of them are targeting the cysteine amino acid in human proteins, as cysteines under physiological pH do have some really nice nucleophilicity to be targeted by these covalent molecules. However, many of the drugs were discovered by serendipity, so having a high throughput proteome-wide platform is really key to discovering new protein ligand interactions, because you want to profile or screen thousands of different compounds across thousands of human proteins. So this whole field or activity-based protein profiling started about 15 years ago, pioneered by Ben Cravat at Scripps, and rapidly evolving in the last 5 to 10 years. And 3 years ago, my former colleague Miliang, who really amazingly incorporated TMT 11-plex with ABPP, so that we were able to work with 48 well plate using probably 50 to 100 microgram protein input, whether these are native cell lysate or live cells, right? So the idea is pretty straightforward. So we treat these samples with DMSO control or one of the electrophiles, right? Then supposedly if electrophile can nicely fit in the pocket and bind to one of the thiol group on the cysteine, you will cover or cap this cysteine residue, and the rest of the reactive cysteines will be captured by using this diethylbiotin-based aldoacetamide probe, which we can further enrich, pull down with strep-avidin microspecies. So he was able to access about 10,000 reactive cysteines from one shot of this experiment, which was then a huge improvement already. But so what we are actually looking for is a reduction in signal in the compound treated samples, right? So that's a competition ratio, we call it. And if you have a CR value greater than 2, we consider it as probably a engagement event, right? And the question for us, or challenge for us, is always, can we get deeper with higher throughput, right? You may think that 10,000 reactive cysteines is a bigger number, but if you consider how many cysteines are in the human proteome, we're barely scratching the surface here. So based on this nice paper, that there are probably 260,000 human cysteines, and we are barely scratching probably 10,000 here and there. So that highlights the challenges or the room for improvement, right? And also, instead of 48 well plate, can we do 96 well plate? Because ideally, when you do high throughput screening, it's going to be 96 well or even 384 well plates. So last year, we kind of overhauled the entire process and came up with this new pipeline that really allow us to work in 96 well plate using probably 10 microgram protein input. We made several critical changes and also incorporated TMT 18-plex based sample multiplexing. The general workflow stayed the same, but now we can do as little as 10 microgram protein input. As a test run or example, we screened about 192 compounds. These are mostly chlorocenamide, and a third of it is acrylamide. They are just common warheads people use in covalent drug screening. And we're reproducibly measuring about 24,000 human cysteines across all the 18 plexes we had. And these are from about 7,000 human proteins in the human proteome. And with all this AlphaFold 2 or 3 that can model or predict protein structures, we're really interested in kind of combine both and see if our platform is really unbiased we want to be able to interrogate or access all the human cysteines instead of just having a biased measurement on a subgroup of them, right? So what you are saying here is the gray histogram represents all the cysteines in human proteome. We're looking at PPSE score, which represents their accessibility based on AlphaFold2 predicate structure, right? So a higher this PPSE score, the less accessible the cysteine is in the protein structure. So what you're saying here is that the gray and the red overlaps perfectly. The gray is all the human cysteines. The red is other those that we can access, we can measure. These are considered reactive cysteines. And the blue are the ones that actually have a good ligand that can bind to it that has a CR value greater than 2, right? So you can further break them down into different secondary structures. The red and gray always overlap, but you do see a slight bias on the blue bars, which means that the molecules we screen do sometimes prefer surface exposed cysteines, which makes sense because they're relatively easier to be targeted, right? But overall, our workflow provides unbiased assessment of reactive cysteines. So we modified the pipeline, the workflow, and we also, with different advancement in MS instrumentation, we are able to get 30,000 reactive cysteines nowadays. So the first paper published by Millian was on the Orbitrap Eclipse that was accessing about 10,000 cysteines. Later, we used the same instrument, but completely changed the workflow. We're getting about 20,000 per single shot. And everybody knows there's Orbitrap Astro on the market. We tried that too. So we are able to profile about 30,000 reactive cysteines from a single shot experiment with a quarter of the starting material that we usually needed for this point here. So yeah, Eclipse Astro 20,000, 30,000. You can also shorten the instrument time. We still get pretty decent number there. So it's 50% higher number and also 25% of the proteome input. We profile three most commonly known promiscuous molecules, KBO2, O3, O5 in those response curves and two replicates. With Astro, we got 31,000 cysteines quantified, and that's about 17,000 on the Eclipse, which we published in our previous paper. And between the two replicates, we have a 95% overlap, which is really good reproducibility there. And you just see these nice dose response curves, and also many of the cysteines we quantified for the first time. We never saw them in our previous profiling. And then we can also use it as a way to identify drug targets. Sotorasib and adagrasib, right, are two FDA-approved KRAS G12C inhibitors, and KRAS G12C are a common mutation in many different types of cancer, lung cancer, colon cancer, and ARS-1620 is a experimental molecule that targets the same site. So we see nice engagement on KRAS G12C in two different cell lines, and also at higher dose, you do see some off-target effect, but you don't have to worry about it because you have to use micromolar, which is not really going to be used in real patients. This is adenosine kinase cysteine 140. You see those response effect too with adagrasib in both of the cell lines. And we can cover a wider concentration range to get the dose response curve as well for the design targets. You get all this nanomolar IC50 values too. So everything looks amazing, right? What are the challenges that we need to address to really make it perfect, right? The first challenge I would say is missing values in this kind of discovery global TMT ABPB screening. So these are three categories of highly valuable drug target proteins, right? Transferrin factors, protein kinases, ubiquitin ligases, and they tend to exist in lower abundance than other proteins like cytoskeleton or histone, right? And when you perform a drug screening, because the stochasticity in shotgun method, you always have this missing values. So here I'm showing you screening, a small screening across 3 different cell lines, and you can appreciate the missing values here, right? It's a huge amount of missing value. It's not gonna be ideal if you're trying to screen thousands of compounds against these drug targets, right? So missing values make drug discovery against valuable targets difficult. You can't afford to rescreen them because there are so many missing values. There's so much resource needed. And again, another challenge is suboptimal accuracy with MS2-based TMT quantification. So we published this data in our recent paper, but basically we created this 2 proteome model by spiking at 1% or 10% of yeast into human at 1 to 1.5 and to 2 ratios, right? So the human background is always constant. So we're expecting to measure a 1 to 1.5 ratio, but because this co-isolated interference that compressed the ratio down towards 1 to 1, what we are actually measuring is 1 to 1.3 on the astral. And as a result, only 50% of these yeast proteins are called significant changes. We're expecting 100% to be significant, but we're only getting 50% of them here. However, if we do MF3-based quantification, especially with the real-time search-based MF3 on the Tribal system using Eclipse, we are measuring 1.5 as we expect 1.5 there, and we're getting 90% yeast proteins called significant changes. So I guess it's a balance between more identifications versus accurate quantification. And remember, we have 5 replicates here in this sample per group or per condition. You don't expect to have 5 replicates when you do a large-scale drug screening, right? And again, this is about precision. Precision is also lower comparing TMT on the astral versus RTS-based MF3 strategy on the Eclipse. And it's about 10%. Across 5 replicates, it's about 6%. And for the lower abundant ones, we can put them into 4 different quantiles. And for the lower abundant ones, the CU values is much higher than the RTS MF3-based approach. So suboptimal accuracy and precision make it challenging to screen with fewer replicates. And the last challenge we're dealing with is how we can differentiate engagement from degradation, right? So if you remember the workflow, we start with all this treated samples and peptides. We only enrich for those DBA label peptide, and we discard all the other peptide that represent protein abundance. So what we are trying to measure is this engagement, where protein abundance stays the same and cysteine is decreasing, right? That's engagement. But what could be also happening is just purely degradation. Protein decreases and the result cysteine decreases, but we don't have this part of the information. That's something we're missing out here. So this can lead to false positives in like discovery screening. What we come up with is trying to incorporate or recently developed real-time target proteomics workflow, which we call GODIC. So basically, GODIC tries to make decisions in real time to calibrate retention time, to detect peptides, and also identifying them, followed by MF3-based quantification. So it's really accurate and sensitive, can do single-shot target pathway proteomics. That's about on average 6 minutes per sample. We don't need to do any sample fractionation. So this was a benchmark experiment we published where we spent 36 hours to measure 10,000 expressed proteins in these 4 human cell lines, right? And then we start to randomly pick 20 groups of any 200 proteins and put on the target list for GODIC and go after them with 2-hour measurements. And at the end, we are able to show that we can measure 94% of these target proteins no matter what the targets are. So we think this really has great potential to enabling large-scale compound screening without missing all those critical information I just mentioned. Yeah, if you remember, this is the traditional ABPP TMT workflow. You enrich, you lose all the protein abundance information, and you measure with traditional DDA that only bias towards the most abundant peptides. However, nowadays with Gold, or we call it cystic now, we want to make an enrichment-free target chemoproteomics platform that we can measure both engagement as well as protein abundance in a single assay. This is a small-scale screening we did. It's about 288 compounds in live K562 cells. It's really standard bottom proteomics workflow because there's no enrichment anymore. What we are targeting with cystic is cysteine-containing peptides and also corresponding non-cysteine peptides that represent protein abundance. As there's no enrichment, we can create a sample that's probably 100 microgram total material that allows us to do 100 times screening. So the first screening we did is we target 300 peptides with functionally annotated cysteines. These are enzymatic sites or structural cysteines that are proved to be important. We screened like 288 compounds with duplicate measurement, 3-hour incubation that give us 36 TMT Pro 18 plexes. So we benchmarked it against on-target approach, just discovery, ABPP, and you can appreciate the missing values. It's a massive amount of missing values in discovery ABPP versus 94% data components using cystic. And we found this many cysteines that have at least one good ligand that can bind 75% of it. And here are two different examples to show you degradation versus engagement here. So in the same assay, we have one peptide that contains the cysteine, the target cysteine, and also a couple other peptides from the same protein to represent the protein abundance, right? So this is a protein CKB. You can see that on the axis level is cysteine peptide level, right? So you see the cysteine peptide decreases, whereas on the y-axis is the protein abundance. You see the protein abundance stays constant, which means this is a purely engagement event, and this is one of the compound that can dramatically engage the cysteine. On the other hand, we do have other examples. We see both degradation and engagement, right? So in this case, you see cysteine peptide level decrease and also protein level decrease. So hypothesis could be this binding, this compound binding to this protein actually induces its degradation. So from this kind of screening, we identify a compound E2913 that binds to the cysteine 4341 on the E3 ubiquitin protein ligase HUE1. HUE1 is responsible for transferring ubiquitin from E2 to the substrate and induces degradation through the proteome system. Has several known substrates and is involved in a wide variety of biological processes. We screened it and we got no missing value, measured every single compound, and identified this one here that can engage. This is probably 70% of the cysteine. And then in addition to measuring abundance and engagement, we can also measure some substrate abundance to learn some cool biology there. So we treat the cells with this compound again at different concentration, bottom proteomics workflow, combine sample, measure cysteine occupancy, protein abundance, and substrate abundance. Because the hypothesis was if we can bind to the cysteine, which is active site cysteine, right, that should abolish its bioactivity, so all the substrates will no longer get degraded. So with increasing concentration, we see increase in the occupancy, and also as we expected, we inhibit the E3 ligase activity. We're seeing increasing abundance of the substrates because they are no longer getting degraded. And then we can go back to the global profiling or TMT BPP workflow to really study the selectivity of this particular molecule. We profile about 24,000 reactive cysteines across the human proteome, and E2913, I believe, will bind to like a couple dozen of them, and HUE1 cysteine 4341 is one of them. And we also luckily found the IDAC ion on an active site, so this peptide has that E42913 has the modification. It's a covalent modification. So to wrap up, I hope I showed you that TMT-ABBV allows global assessment of protein small molecule interactions for 20,000 cysteines using as little as 10 micron proteome input on the arbitrary eclipse. And advancement in MS instrumentation enables measuring 30,000 cysteines on the astral, and TMT quantification using RTS-MS3 provide the best precision, improved accuracy, and greater success rate in detecting significant changes, especially when you're talking about using fewer replicates for large-scale screening. And target proteomics using intelligent data acquisition platform Gold can flexibly create powerful high-throughput assays for ABBV screening, and cystic is enrichment-free and can simultaneously measure engagement and protein abundance to reduce false positives in ABBV screening. So without that, I want to thank all the people who have contributed to the workflow I showed you today, people from my lab and also my amazing postdoc mentor, Steve Key, and folks in his lab too, collaborators. And yeah, if you are interested in learning more about lab, please check our website here. Thank you. Happy to take questions.
Thank you so much. We will now turn things over to Alexey.
Hi everyone, my name is Alexey Chernobrovkin. I'm principal scientist at Pelago Bioscience, and in today's presentation I'll tell you about target deconvolution based on cellular thermal shift assay and different quantitative proteomics approaches we are using for it. A few words about our company. Pelago Bioscience is a CRO located in the beautiful Stockholm, Sweden. CETSA is our core technology, which we are using to help our clients in their drug development programs. With more than 12 years expertise, we have more than 250 customers worldwide, including top 10 big pharma companies. CETSA stands for Cellular Thermal Shift Assay. Unlike classic thermal shift assay used for measuring purified proteins in vitro, like DSF, CETSA is run in a native cellular environment for measuring target engagement inside the living cells, closer to biophysiological conditions. No modifications of the protein or the compound is needed to run the assay. And the principle is, if the compound binds to your target protein, it will change its thermal stability and result in shift in melting curve to the right if protein got stabilized, or to the left if it was unstabilized. Thus, by measuring protein denaturation curves inside the living cells, we can deduce whether protein is bound to the compound or not. CETSA can be applied for target deconvolution studies in different ways. In 2012, Mish Savitsky combined isobaric labeling quantitative proteomics with CETSA, enabling thermal proteome profiling. The method allows identification of compound-induced thermal stability changes for thousands of proteins simultaneously. While being a very versatile and powerful tool, there are several challenges in applying TPP for real-life target deconvolution problems. For instance, the number of samples need to be analyzed is quite high, given the 10 temperatures and 2 conditions, and 2 replicate easily results in 40 samples to be processed. Also, proteins need to be quantified across very different backgrounds. Proteomes, after heating to 37 degrees and 70 degrees, are very different ones. In 2018, Roman Zubrev group published PISA approach, where some of these challenges are being addressed. In PISA, melt curves are being integrated in vitro by pooling samples together after the heating step. In such way, soluble protein amount becomes proportional to the area under the protein melting curve. Comparison of soluble protein levels at different treatment conditions allow identification of compound-induced stability changes in a much more efficient way. However, this approach also poses few challenges. For instance, observed compound-induced changes can be driven not only by changes in protein stability, but also by changes in solubility, protein abundance differences due to the incubation, or extractability given the specific protein extraction condition. Another challenge here is that the fold changes in this approach, they're relatively low, and in order to resolve such low fold changes, one needs to have really high precision quantification. Isobaric labeling using TMT helps to address some of the challenges of drug target deconvolution strategies. High multiplexing capability, excellent quantification precision, and dim protein coverage makes it an indispensable tool for target deconvolution using SATSA. To highlight power of TMT-based SATSA profiling, we've decided to investigate natural product allantolectone. Over the last few years, it drew lots of attention in literature due to the various potential therapeutic effects, including anti-cancer and anti-inflammatory effects observed in vitro. Though several papers have been published already, there is no clear mechanism of action discovered for this compound yet. We've also used this opportunity to evaluate recently released 32-plex reagent from Thermo, where original TMT Pro 18-plex capability was expanded by adding a set of 17 deuterated tags to the original 18-plex. On the bottom of this slide, you can see the reporter ions for this 32-plex, and you can see that the peaks are closer to each other compared to the standard 18-plex, and as a result you need twice as much resolving power to be able to resolve all these reporter peaks. One potential problem which may arise while using the 32-plex is that deuterated and non-deuterated peptides may have slight shift in retention time, which can be mitigated by adopting the experiment design to focus on the all statistical evaluation be performed within either deuterated or non-deuterated set, or having some bridge channel across the deuterated and non-deuterated sets. To test different ways how 32-plex can be used for efficient compound profiling, we've tried two approaches. We've started with intact cells and lysate treated with different concentration of allantolectone. So here you can see 4 different concentration of allantolectone plus DMSO control. Treated samples were aliquoted for heating to different temperatures. So on this slide we can see 8 temperature points. And then there is 2 ways how to proceed from this point. We could either pull them together in the temperature dimension to generate PISA set, what we've done for both intact cells and a lysate, resulting in 32 samples, which were labeled together with one set of 32-plex reagent. We can also choose one replicate per concentration, choose 3 different concentration and DMSO control, and take individual temperature points, and again label them with 32-plex reagent, resulting in 32 samples, and analyze those. So the ones to the right will represent more classical TVP approach, while the ones to the left will represent PISA approach, where intact cells and lysate experiments combined together. After labeling of the peptides, peptides were separated using high pH fractionation into 60 fractions, and every fraction was analyzed using Orbitrap assay and mass spectrometer, applying the real-time search SPSMS3 method with 45 million gradient per fraction. This resulted in pretty deep proteomic profiling with more than 7,500 proteins being quantified. If we look at the quantification precision, we can evaluate the median CV of the proteins in lysate and intact cell experiment, and can see that for the lysate we have around 6% median CV, while in intact cell we have slightly higher CV, up to 9%. For the melt curve experiment with the TPP, we didn't calculate any CV because we only had one replicate per condition. If we look at the results of the first experiment, when we use PISA approach and profile compound in intact and lyse cells with 4 different concentration parametrics, we can see that a number of proteins were detected with compound-induced change in thermal stability. So here the results presented as volcano plots when the y-axis correspond to the significance of observed compound-induced change and x-axis correspond to the thermal stability difference. Positive values correspond to increase of stability, negative values correspond to the decrease of stability. And in the middle, you can see the scatter plot of thermal stability changes detected in lysate and intact cells. What we can see here is that there is a set of proteins which behave exactly the same between intact cells and the lysate, but also there are some thermal stability shifts which are more predominant in either lysate or intact cell experiment. Having multiple concentrations in one experiment is very useful when you want to profile unknown compounds, and here to the right we can see concentration response profiles for selected significant protein hits, and we can see, for instance, for proteins like AKR1, C1, C2, C3, we have almost overlap concentration response in both intact cells and lysate, or for some proteins like AKR A1 and B1, we see slightly lower concentration response in the lysate compared to the intact cell. And we can also see that a number of proteins were found to be destabilized, for instance batch 1 and KEAP1 being destabilized in lysate. Batch 1 was also found to be destabilized in intact cell as well. Interestingly enough, that both KEAP1 and batch 1 are key enzymes in oxidative stress response, with KEAP1 being part of E3 ubiquitin ligase and batch 1 being transcriptional regulator. So these results here show how 32-plex can be efficiently applied to profile one compound in multiple matrices in multiple concentration in one go, providing you with a plethora of useful information. If we look at the second experiment result, when we profile compound just at 3 different concentration plus control, but having all 8 temperature point present, we can use this to plot the individual melting curves for the proteins we select. And that gives us totally another dimension to the data. For instance, we do see here proteins like AKR1A1 or PTGR2, which behave exactly textbook SATSA examples when you have compound concentration dependent increase in stability. For PTGR2, you see gradual increase at all concentration tested. For AKR1A1, you can see overlap melt curves at 0 and 1 micromolar concentrations, and also fully stabilized curve at 10 micromolars and 30 micromolars. What is interesting to see is that for some proteins, some other proteins like AKR1, C1, C2, C3, we not only see the stability changes, which is can be visualized by right or left shift of the melt curves, we also see shift in the melt curve position along the y-axis, which correspond to the increased amount of the protein available already at the lowest temperature applied, 37 degrees here. So we can see that some of the changes which were observed in the PISA experiment driven not only and not mainly by the stability change, but also due to the change in protein solubility. In some proteins like KEAP1, we don't see any changes in protein melting temperature, but all the difference, apart from destabilization, which we see, it comes from the change in soluble protein level. Similar is for the DHFR, when we see both right shift of the melt curve, but also up shift on the melt curve. So it's combined effect, increase in stability and solubility or abundance. Taking together this data set, which was done with 2 × 32 plex, not only given us potential drug targets of allantolectone in cells and lysate, but also showed us how this compound affects all these individual proteins in terms of the change in stability, solubility, or protein abundance. In the example showed above, we've used TMT to do SATSA profiling of the compounds. And even though the multiplexing capability was increased up to 32, it still takes up to a couple of days in order to finish the data acquisition. So we basically have the throughput of 16 to 32 samples per day. However, in order for us to enable large-scale experiment, generate enough data to be able to use AI, ML, data analysis and interpretation toolset, or, which is important for us as a CRO company, to offer fast turnaround time services to our customer, we need even higher throughput compared to 16 or 32 samples per day. In order to achieve that, we had to combine recent advancement on different steps of the workflow together, starting from sample prep, when we automated as many steps as possible using liquid handler robotics. We also use robust chromatography systems, Vanquish-Neo or EOSEP, working with Aurora columns from Ion Optics, providing us with excellent separation. And of course, we use capabilities of newly launched Orbitrap Astral mass spectrometer, working at the super high speed and data acquisition mode, and recent advances in data analysis and bioinformatics, namely Spectronaut 19, which works nicely in both DIA and direct DIA mode. Combining them all together allows us to switch the throughput to up to 100 samples per day and still provide excellent quantitation, depth and accuracy across the samples. If we look here, we can see that every single bar here consists of 96 smaller bars. So basically it's one plate, one plate, one plate, one plate, and so on. On the top plot, we see a number of proteins quantified in experiments, and we can see that we routinely stay above 6,000 proteins across the whole plate. And in the bottom plot, we can see median CV across the triplicates, and we see that we're routinely below 6% in terms of the median CV. So we can achieve extremely nice reproducibility from plate to plate, from month to month, running samples at 100 samples per day, which translates into 196 plate per day, quantifying more than 6,000 proteins and able to profile up to 30 compounds and triplicates per plate with a good proteome coverage and great quantification precision. What can we get with this kind of data? For instance, we can profile a library of compounds and see similarities or differences between the compounds of interest. On this slide, we focus on the HDAC inhibitors, and we can look at the data from different angles. On the left plot, we can see multiple compounds at the x-axis, and the y-axis correspond to the thermal stability shift. And we see 3 proteins, HDAC1, HDAC2, and HDAC6. We can see the compounds which cause significant change in thermal stability of HDAC1, HDAC2, or HDAC6 on each of this plot, and we can already here spot some key differences. For instance, entinostat stabilize HDAC1, HDAC2, but doesn't stabilize HDAC6. And the opposite, droxinostat stabilize HDAC6, but not HDAC2, and to some extent HDAC1. We can turn around and plot proteins as an axis and then color code different compounds, in this case 3 HDAC inhibitor being color coded, and that also allows us to spot some differences and similarities between these different HDAC inhibitors. If we look, for instance, for HDAC1, we can see similar thermostability shifts by panobinostat in green and belinostat in blue, but much lower for droxinostat in red. If we look at the TTC38, which has been previously annotated as an off-target of panobinostat, we can see it's exclusively stabilized by panobinostat and none other HDAC inhibitors. And if we look at the LAP3, which also non-off-target of panobinostat, we can see that it's been stabilized into the same way with all 3 HDAC inhibitors. So that gives you different perspective, depends on the question in mind you have to the data. And now we offer into our clients selectivity profiling service, in which we profile compound in standardized cell lysate using automated workflow and high-throughput DA-based protein quantification with a streamlined QC, easy-to-interpret report, and we deliver all of it just within 10 days from submission of the compound. A few more examples what it can be used for and how the data can be viewed. For instance, here we profile 2 known CDK4/6 inhibitors, abemaciclib to the left and palbociclib to the right. We can clearly see stabilization of primary targets CDK6 and CDK4 by both compounds, as shown here. We can also see some off-target kinases being stabilized, in this case by abemaciclib. But if we include these compounds at different concentration into this profiling, then we can also look at the data in a different way. So we can look at the concentration responses of the primary targets in different compounds, and we can see very similar response pattern for 2 CDK4/6 inhibitors. We can also see the cyclin associated with this CDK4/6 also being stabilized in this experiment. We can also see some off-target kinases, mainly specific for abemaciclib in this case. We also see some off-target kinases, and here in this case it's specific for palbociclib and not for abemaciclib. We see non-kinase, in this case specific for abemaciclib, nothing happens with the palbociclib. And we can do it in a very efficient and reproducible way. Also, when we have lots of data accumulated for the compounds in the same way, then we can start to use AI or ML tools to answer some other questions. So we can look for compound similarity in terms of the thermostability shift profile. And we can see, of course, that we have clusters of HDAC inhibitors, DNA intercalators, microtubule inhibitors, kinase inhibitors, NSAIDs, and so on. With all that, I hope I managed to convince you that precise and efficient protein quantification is essential for drug target deconvolution, that increased plexing capability of 32 plex enable more comprehensive experiment design, which gives you more opportunities to investigate your drug in more details, and next generation methods and instrumentation dramatically increase throughput and data quality in drug target deconvolution studies. With that, I want to thank you for listening to this webinar, and also want to thank colleagues from Thermo for allowing me to present here, and especially I want to thank Albert Arl for producing this data using Orbitrap Ascent in a very fast and efficient way. Thanks everyone, and happy to take any questions.
All right, thank you so much, Alexey and Qing. We are now going to move into our live Q&A portion of the presentation. So if you've not done so already, please use that Ask a Question box just below this presentation to ask your questions. Alexey and Qing, if you would like to come online now. There they are. And we'll begin with our first question. First, some kudos saying awesome work. Is there an associated publication I could reference for the SysDig platform workflow?
Thanks for the question. And so we are working on publishing it there isn't one yet, but hopefully soon.
Right, fantastic. Our next question, what stage of the drug discovery pipeline are CETSA experiments most frequently used? Compound library screening or hit validation or mechanism of action?
Thanks for a great question. So typically when we're talking about the proteome-wide studies, when we use CETSA for target convolution, then it typically happens at the later stages, or it could be done with the hits from phenotypic screens. However, in general, CETSA can be applied even for the primary screen, especially for the proteins, which is hard to establish the functional assay for. In this case, we can set up high throughput CETSA assay based on fluorescence or other type of detection, and it can be used in the primary screen.
All right, fantastic. Another question here, what drug discovery projects are better suited to use DIA or TMT with CETSA experiments?
Thanks again for the question. So the TMT provides you with the exceptional quantification precision, and you can go as deep as you have time for into the proteome. So if you have just one, two, or a handful of compounds and you want to know most information about them, I would go with TMT. However, if you want to go wide and investigate a series of compounds or a small library of the compounds, then the throughput of DIA shines, and I would go with the DIA with that.
All right, thank you so much. This next question is regards to the last slide. So I'm not sure if we were talking about Qing's presentation or Alexey's, but you two will be able to decipher that. It's a longer question. In your last slide, you were able to detect the ligand-modified cysteine peptide. Was this from a targeted GoDig method, TMT, or DDA method? And then I do have a part 2 to that. So if you'd like me to break that up into 2 parts.
Yeah, I can answer the first part. I mean, it's not from, it is from the regular bottom proteomics, and that's where we are all expressing that protein to see that ADAC ion. So it's not from a standard system profiling.
All right. And then our second part, why is it so much more successful to measure competition ratio and decrease of peptides and not detecting increase of ligand-bound peptide?
I mean, it's successful and much easier to do that because you know the modification, which is DBA probe. When you do the down assay, we're measuring that reduction in signal. Versus if you want to measure the increase the compound bond form, you have to figure out the delta mass for every single molecule screen, right? There might be weird things happening on the mass spec or during sample prep that add other modifications to your compound too. So it's going to be a lot of work, but it's doable. And yeah, using DBAs makes things easier to screen hundreds of solid molecules. Yeah.
Right. Thank you for that. The next question, you described the missing value challenge for ABPP and showed how GoDig can address this for a targeted set of peptides. Would increased fractionation, and then it's an acronym FAIMS or FAMES, or DIA help with missing values too?
Yeah, I guess a question for me. Yes, totally. If you have the bandwidth and instrument time to run many more fractions, it can solve your problem. I just don't know how much more time you can afford to run those fractions, right? If you're going through many samples. Yeah, it's a balance between the throughput and like how deep you want to go with just regular discovery, untargeted approach.
All right, thank you so much, Qing. Our next question, does the discovery of additional off-target proteins for a drug in a CETSA experiment explain drugs with more harmful side effects? How does finding surprising proteins result in these experiments affect the drug discovery process?
That's a great question. To our experience, there are several proteins which binding to which could suggest several potential side effects like phospholipidosis or something else. There are other anecdotal evidences when binding of the compound to a specific target can cause very severe side effects, like thalidomide binding to E3 ligase cereblon cause the degradation of several transcription factors, result in severe defects from the when it's used during the pregnancy. And again, these kind of events, they can be started during the study of target engagement and binding specificity profiling of the compounds.
Right. Thank you for that. And our next question, does TMT give an insight at treating or help in curing genetic-based disorders?
Well, that's a really interesting question there. I can give a shot first. I think, yeah, if you're talking about, because TMT helps measuring proteins, right? If you're talking about there's some malfunction of protein due to a genetic disorder, yes, it can help with that. But if you're talking about directly, for example, CRISPR or anything else, genetic therapy that directly goes after genes, there's not much for TMT there, because TMT is for proteins. Yeah.
All right. And Alexey, did you want to touch on that? I apologize if I cut you off as you were going into your answer as well.
No worries. No, I agree that, I mean, TMT is wonderful technology to quantify proteins, to perform relative protein quantification. And if proteomics help you to decipher the molecular mechanism leading to the molecular phenotype of genetical disorder, then we could find potential protein targets to mitigate those issues for sure.
Right. Fantastic. This one is for Dr. Yu. Could you provide more details on how you came up with the inclusion lists for the user-defined lists of peptides? For example, was it based on prior experiments or in, I believe it's silico digestions?
Yeah, it's a good question. So we do prefer like prior data from prior experiments, so we know they're expressed and they are detectable, right? And there's, because there are just millions of peptides, you never know how good they fly in your mass spec. But we also showed we can do predicted peptide or spectra too. Then you have to curate or optimize that included list to make sure those peptides are good for your specific acid or target acid. Yeah.
All right, wonderful. All right, we're getting quite a few questions. I really appreciate the questions that are coming through. We're going to get through as many as we can in the next couple of minutes. Our next question asks, does the discovery of additional off-target proteins for a drug in CETSA experiment explain drugs with more harmful side effects? How does finding surprising protein results in these experiments affect the drug discovery process?
I think we already answered this question.
Oh, we did already. Apologies. My apologies for that. Our next question then, when you see a larger thermal shift in the cell versus lysate, do you attribute that to protein-protein interactions and protein complexes or spatial protein location? And there is a second part to this as well.
Yeah, so I mean, the size of the shift which we see in this or that matrix depends on the initial stability of the proteins and the change in stability caused with the compound. And of course, there could be difference between the protein state inside the cells and in the lysate. So it's hard to interpret, but in some cases we can interpret it as the change in the protein-protein interaction state between different cell stages or cell and lysate.
All right, fantastic. And the second part to that question, have you tried to follow up with spatial proteomic experiments or protein-protein interaction studies?
Yes, we tried few approaches to follow up on the sets of studies. And basically, what we do with the thermostability changes is we detect the events of the initial binding, and then we can also see the immediate reactions within the cells, like the signaling propagation. And then we can also see the protein translocation change in protein stability. And those events we can trace further on by following the protein location within the cells and see how this binding effect leads to the change in the protein location within the cells and the change the cell morphology.
All right, I'm going to sneak one more in. Are there other reactive amino acids that could be screened with ABPP as you showed for cysteine?
Yeah, there are a bunch of others that people try and show it could work. For example, lysine, tyrosine, and even histidine. I believe there are people making different kind of probes. You just have to need a really good probe to kind of access all those amino acids.
All right, fantastic. So with that, I am going to thank Qing and Alexey again for their time today and for your important research. We'd also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank you, our audience, for joining us today and for your great questions. Any questions that we didn't have time for today or any that are submitted via the on-demand period, those will be addressed by the speaker via the contact information that you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay, and we encourage you to share that out with your colleagues who may have missed today's live event. Until then, again, take care, everyone, and thank you so much. Have a great day. Bye-bye.
Thank you. Bye-bye.